Good morning, students. This is Mr. Russell. I'm here for the second installment of your notes for Objective 5 about DNA and replication. We already talked about DNA, and today we're going to talk about chromosomes, which is big chunks of DNA. Okay? Just a little review. Remember that DNA is the instructions for making living things. All the different proteins that your cells make that make you, and not a cockroach, all come from your DNA. And it consists of two strands, which you can see right here. The sides of those strands are made up of sugars and phosphates. Okay, they don't really hold any information. They're just there to hold the important part, the bases, together. And in the middle here, you have your bases. There's 4 of them: A and T, C and G, which we're going to talk about in a lot more detail later. That stands for adenine, thymine, guanine. Oh no, sorry, that's cytosine, and this is guanine. Okay, and that makes up your DNA. And you have these individual subunits that are like links in a chain, and those are called nucleotides. So that's a sugar, a base, and a phosphate. That's one nucleotide. This is another nucleotide, and so on and so forth. The 2 of them put together across from one another, this is called a base pair. So these are matching nucleotides. And your DNA in every single cell in your body, you have about 3 billion, with a B, billion base pairs. All right, so let's talk about chromosomes. So chromosomes are big chunks of DNA that are divided into separate sections. I like to think of them as like chapters in a book. You have a lot of sentences made out of words and letters in a book, but that's broken up into chapters so that it's easier to read and easier to work with. Okay, we refer to these things as being supercoiled. I'm sure you know what a coil is. But supercoiling, let me see if I have a rubber band and I can show you. Supercoiling is when something coils, like you see here, and then if you keep coiling it, it will eventually, the coils will start to coil up on themselves, and you end up with something that looks a little more like that, and then it'll continue coiling. I can't coil this one very far because it's a very short rubber band. But that's supercoiling, and that means coils that get coiled, and what it is, it's a very, very tight, dense way to store DNA. Kind of like wrapping thread around a spool, okay? You might have hundreds of or thousands of yards of thread on a spool, but you don't want to have to deal with hundreds or thousands of yards, so you wrap it up around that spool, and it stores it very efficiently. Okay, and this is kind of what it looks like. We have our double-stranded DNA over here, and then we have our DNA threaded around these histone proteins, which are kind of acts as the spools, and then that in itself gets coiled again, and then again, and then again, until you end up with an actual chromosome. So they're very, very tightly compacted. Okay. Each chromosome duplicates in preparation for mitosis. Mitosis we're going to talk about in our last objective this semester in December, and it is the process of how cells replicate themselves. It's how we grow. You're made out of trillions of cells that all started from one, and they have to divide in order for you to grow. So part of that process is DNA copying itself. That's called replication or duplication. Okay, so as part of the process of replication, you end up with sister chromatids, and these are 2 exact copies of the same chromosome. And you need those exact copies so that when you make 2 new cells, both cells get a new copy. So you'll see this on your notes. 10A says after replication blank blank remain attached. The sister chromatids remain attached at the centromere is what goes in 10A. Okay. Chromatids are a single strand of a replicated chromosome. So that's 1 copy of 1 chromosome. And the reason we point out single there is because you are eventually going to end up with 2. All right, and the centromere is a place on those chromatids where they anchor together. All right, and this is what you'll see here. This is one chromosome. We would also refer to it as 1 chromatid, but after it replicates, you'll notice we have twice as much. Well, this whole thing now is referred to as 1 chromosome because it's 2 copies of the exact same thing. And this side of it is a chromatid, and this is the other chromatid. And these are sisters because they are genetically identical. And this little pinched spot here in the middle is the centromere where the 2 of them join. Okay, Let's talk about chromosome number. All right, a chromosome number is an organism's total number of chromosomes, and it is definitely not the same for all organisms. All right, in humans, you have a total of 46 chromosomes. You may have heard that before, and those come in 23 pairs. And what that means is that you got 23 from your mom and 23 from your dad, and together those pairs equal the number 46. And I kind of think of them as shoes. Imagine that every time you got a pair of shoes, your parents went to the store together, and mom bought you the left shoe, and dad bought you the right shoe. So you always have the correct number of pairs. And hopefully they coordinate so that one buys the left and one buys the right, so you don't end up with a bunch of 2 lefts and 2 rights, because that would ruin the analogy. Okay, fruit flies have far fewer chromosomes. They have a total of 8 chromosomes for a number of, I guess the pair number, the N number is 4. So 4 pairs for 8 total chromosomes. All right, pea plants, 7 pairs for a total of 14. Okay, elephants have more than humans do. People tend to assume that the bigger and more complex an organism is, the more chromosomes it will have. That's not always the case. Obviously, elephants and humans are bigger and more complex than pea plants and fruit flies. But actually, there's some species of butterfly that have 250 chromosomes, and they're definitely not as complex as humans are. So, a lot of times, smaller, simple organisms have less chromosomes, but that's not necessarily the case. Okay? Remember, your chromosomes exist in pairs, 1 from each parent. It's 23 pairs for humans. And when they exist in pairs, we call that diploid. Remember, this prefix di means 2. And the ploid just refers to chromosomes. You'll see that pop up now and again. Okay? If we're talking about a single set of those, like what you would find in a sperm or an egg, because that's before they've joined, so the sperm from the father, the egg from the mother, we call that haploid. And the easy way to remember that is just half. It's half the pair. So one set of chromosomes is haploid. That's 12B and C on your notes. Okay. Moving along. Oh, here we have our diploid pairs. You'll see there's 2 of each. All right. Normally they're not color-coded in your cells. That's just to make it a little easier to understand. And here they're haploid. You have 1 of each. So it's kind of opposite of what you see right here. Okay. Let's talk about male versus female chromosomes. Okay. They are going to be exactly the same with one difference. A female will have 2 X's. So she gets an X chromosome from mom and an X chromosome from dad. All these other ones, these other 22 pairs, are going to be the same. These are slightly different diagrams just because they came from different sources. That's not a difference between men and women. And then down here, you'll see that men have an X and a Y. So that means they got an X from mom, but dad gave them a Y, and that's how gender is decided in sexual reproducers. The female always gives an X, and then the male has a 50-50 chance, since he's got an X and a Y anyway, he's got a 50-50 chance of giving an X or a Y chromosome. If an X, you'll end up with a baby girl, and if it's a Y, you'll end up with a baby boy. Okay, so these pictures here we're going to talk about on the next slide. These are a picture of someone's chromosomes, and we call those a karyotype. When you start out, you end up with just a mess of chromosomes like this. They actually will use a tiny syringe and suck them out of the nucleus of a cell, and then you kind of lay them out flat on a microscope slide, and you take a picture, and then with the picture, you cut them up and you match these pairs. Okay, and this one's kind of a fun neon colored one that somebody used dyes to make them that color. Or more likely, they just edited the image actually. Okay, so a karyotype is a method of diagnostics, meaning a way to diagnose things. And what it does is it diagnoses certain genetic disorders, what we call chromosomal disorders. All right, a chromosomal disorder is when you have too many or too few chromosomes. Because you might remember humans are supposed to have 46 chromosomes, but sometimes during certain phases of reproduction, you end up with an extra or a missing chromosome. So if you're supposed to have 2 of each pair, if you have 3, we call that a trisomy. So because you have 3 of that chromosome instead of 1. And if you only have 1, we call that a monosomy. Remember, tri means 3 and mono means 1. Okay. Most of the time, these are so serious and so detrimental to the baby that they'll cause a miscarriage. A lot of times it'll happen so early that maybe the mother doesn't even know that she was pregnant. It'll be terminated so early. And some are less serious, meaning that the child can actually be born, but they still cause serious health problems later on. So I have a few examples of those that I'll talk about. Klinefelters is when the X's don't separate, and so you end up with a boy that has 2 X's and a Y chromosome. And here's a karyotype that shows you that, 2 X's and 1 Y. And that involves in some pretty serious cognitive problems, meaning brain function doesn't do exactly what it's supposed to do. Okay, Down syndrome you've heard of, that's a trisomy 21, and that's when they have 3 of the 21st chromosome. Okay, and I'm sure you're familiar with Down syndrome and some of the things that that causes. Patau and Edwards are trisomies of the 13th or 18th chromosome, respectively. And those are both very serious and usually lead to the child dying fairly quickly. I think sometimes they could live to a few years old, but they definitely do not grow to adulthood. And that's because some of the birth defects involve their organs, and if your major organs aren't functioning properly, then you don't have a lot of chance of living a very long life. Turner syndrome is actually probably the least serious health-wise, and this is a monosomy X, so a female that only has one, oh no, I'm sorry, that's the wrong picture. Here we go. Only has 1 X chromosome instead of 2. And from what I've seen, these women actually lead fairly normal lives. It's actually fairly common, not common, but it's not rare. So those are examples of chromosomal disorders. And these, remember the pictures, are called karyotypes. And all these little guys you see on all these things are individual chromosomes, which are just big bunches of DNA. Thanks for watching.